From the University of Toronto's Leslie Dan Faculty of Pharmacy, this is the I'm Pharmacy podcast. I'm Mina Tadrous. In preparation for this podcast, I spoke with a number of people and simply asked them, how do you think we invent new drugs? People consistently answered the same way. You find something that needs replacing and you replace it. And they aren't too far from the truth in many cases. But like we found out in episode 1, there are so many different paths to lead to the same and final point. You can know the final destination or the final target and figure out how to get there, but there's actually a lot of different and uncommon ways to get to the same point of drug discovery. Two important themes that seem to be consistent in a lot of stories are accident and good observation. A great example of this happens on the North American prairies during the 1920s. Farmers would have usually used only specific types of hay to feed their very hungry cattle, but because of the Great Depression, they were using whatever they could get their hands on. Unfortunately, they kept having many of their cattle die of internal bleeding. They would later call this sweet clover disease. Years later, after some good scientists worked on discovering what was actually causing this, they were able to isolate a substance. This was called warfarin. Warfarin was named after the Wisconsin Alumni Research Foundation, the funders for the work. This is a good shout out to anyone wanting to get a drug named after them. Fund some research. From there, they worked to fine-tune this isolate, and they used it for a very important issue: killing rats. Yes, the famous drug warfarin was first used to kill rodents. It wasn't another few years after the discovery of this that it was realized that it would be a great oral anticoagulant and would help replace the injectables that were being used in hospitals to do the same thing. It took more years to figure out how best to use it, how to monitor it, and when to stop it. Warfarin for many years was a cornerstone for anticoagulation. Ask anyone that's been to pharmacy school how important this is. It's been used in everything from post-surgery, those living with cancer, those living with atrial fibrillation, and even most recently, helping those with COVID infection. The story really highlights two things for me. One, how long it may take and how many different steps it goes from isolating something to knowing how to use it, and the improvement that happens. Although today these timelines may be a bit faster, they still do take time. The second is how things build on each other, and how clinical knowledge and targeting can help improve and lay out some interesting questions. And that's what I hope to discover and talk about today. How do we figure out how and where drugs work? Does this ever change? And are there more than just one way to find that final target? To better understand how we find targets, I spoke to one of the world experts in this field, Professor Angers.
My name is Dr. Stèphane Angers. I'm the new director of the Terence Donnelly Center for Cellular and Biomolecular Research in the Faculty of Medicine at the University of Toronto. I'm also a faculty member in the Faculty of Pharmacy and co-director of the PRIME initiative.
Where do you see your work fitting into the life cycle of a drug?
I think our work can be helpful at different levels. Primarily, it is at the very early stage for target identification, right? So elucidating the basis of diseases, what are the genes or the protein products that one should target in order to treat that disease, right? So if you want to treat a disease, you need to identify a couple so-called therapeutic targets. So this is where functional genomics is really powerful to do that, right? So if you can use CRISPR and do a large genome screen, if you will, a genetic screen, to identify, for example, in a cancer cell, which gene, right? The human genome contains 20,000 genes. Which of these 20,000 genes are responsible for the growth of that cancer, right? And we can identify that really well now, very efficiently. The technology is there. In about a month or two, can do these type of experiment. and sift through these 20,000 genes and identify maybe 20 or 25 that are really, really critical for the growth of this cancer cell, right? And the genes that are important for the growth of, let's say, a breast cancer cell are different from the genes that are important for the growth of a pancreatic cancer cell, and so on and so forth. So, if you do these type of experiment at scale, you can come up with a list of targets that are really selective for a cancer of interest, right? So this is an example for cancer, but, this can be applied for any type of human disease. And, there's a few examples that came out of my lab that are now being investigated and translated into the clinic, right? Where we have designed and developed therapeutic antibodies along with colleagues at the University of Toronto. Dev Sidhu at the Donnelly Center is an expert in antibody engineering, and we have partnered with him to develop an antibody molecule, which is now a drug, a potential drug, a therapeutic that, informed by the functional genomic and the target identification, we were able to develop a molecule that then interrupt the function of that gene product. And, so this is the two steps, right? Very early stage target identification. And then there's a molecule or a therapeutic development, which would be then the next step after.
From hearing you speak, it sounds like the world of drug development has changed. When we look back at the old stories, it almost felt like they were finding the molecule first and then figuring it out where it works. Hearing you speak, it sounds like you find where things should work and then find a molecule that should fit there. An old way and a new way. So do you think the new way is the new method forward for all drug development?
Yeah, I think more and more, I would say now across the board, it's very much target identification first, knowing the mechanism of the disease and which genes are involved in a given disease state, right? And then coming up with molecules or biologics or a small molecule that would interrupt or activate the function of a gene product. That's pretty much across the board, I would say, what's going on in the pharmaceutical industry.
And it sounds like most of these drugs being developed are the newer targeted monoclonal antibodies or biologic-based drugs versus the old small molecule type medications. Is that correct?
A small molecule can be also very much targeted, right? It's not because it's an antibody that it's more selective. There are antibodies, of course, that can target multiple gene product. So I think there's maybe a shift in paradigm that has occurred, if you will, over the last 15 years or so. If you think again in the cancer paradigm that we discussed, the best cancer medicine that have been developed over the last 30 years or so were very non-selective, right? Chemotherapeutic drug, they hit the ability of cells to grow basically non-selectively. They don't really attack the problem at the root. What is the issue with the cancer cell? They just non-selectively block the ability of any cell to grow fast, right? And because the cancer cells are the one that divide and multiply the fastest, there's a therapeutic benefit to do that, right? So and this is basically why there is very bad side effects associated with these treatments, right? Like people lose their hair, they have nausea because the hair cells and the lining of the epithelial cells of your gut, of your intestine, are rapidly dividing, right? So using this chemotherapeutic drug lead to very bad side effects. Now, over the last, I would say 15 years, there's been this targeted therapy revolution where rather than just having this naive treatment of blocking rapidly dividing cells, people are starting to use the knowledge that we have about the molecular defects of cancer cells, which genes are driving the growth of cancer. And now much more rationally, if you will, they develop a very selective inhibitor against these, driving oncogene, if you will, right? That are responsible for the growth of cancer. And this way, you can hope to target only the tumor cells, but leave the healthy cells in the body intact, right? Non-targeted. And then the idea is that by being much more selective, you have less side effect, the therapy is more efficient. And there's been tremendous clinical advance on this front, right? The the arsenal, if you wish, to treat cancer, different cancers has completely changed, and the introduction of monoclonal antibodies or other small molecules that are so-called targeted therapy have changed completely the prognostic for a lot of patients.
It sounds like a main theme has been the development of new tools and all the opportunities that it opens up. So do you find yourself, when developing new research ideas and asking questions, starting with the tool first and seeing what questions it can answer? Or do you find the question first and then try to find the tools that can help you answer it?
Both approaches are good, I think, right? So there is a group of scientists that become expert in a technology, right? They are the world expert, let's say, in mass spectrometry, right? And then they're looking for questions to answer with their technology, right? And they collaborate with different groups and do that. The other approach is actually the one that I like the best. Is I come up with the question that I would like to answer first. And then I look out what are the tools and technologies available, what other technology we can come up with and help develop, right, to address the question that is of interest. So I think there's no good or best avenue. I think it's a personal choice of how you want to operate.
So how do you find those questions?
I think that's the beauty of being a scientist, right? You can raise an hypothesis and then you test it with the various tools that you have and the various experimental strategies. Another strategy is just do a massive screening type of exercise. And I think just to go back to the technologies that are available nowadays, it is perfectly fine to do this because it's so powerful, right? So you can actually, rather than say, okay, I think that gene X and Y may be important for this process I'm studying. Why are you not looking at the whole genome at once? All 20,000 genes, right, in the human genome, and ask which ones are important in a very unbiased fashion, right? So this is not hypothesis driven. This is actually, doing a complete survey of the genome, right? So I think more and more with these enabling technologies, it is possible to do that, right? So, the traditional science and scientists that say, okay, you need to raise an hypothesis and test it, that's fine, right? But also, I think more and more now with these type of technology, I think you can be a little bit less smart in a way about it and just do the experiment, right? And then the works begin though, right? Like, what do you do with this information? How do you go about proving that, gene X, for example, is indeed important for the phenotype you're studying, figuring out how it works? There's still a lot of insights that needs to go into this.
From speaking with Dr. André, we can see that advancements in technology have changed the game in disease target identification. In fact, in some cases, it has even flipped the scientific method upside down. It has allowed researchers to go into an experiment without a hypothesis. More than identifying targets, this newer technology has expanded exploration and led to much more targeted therapies with less side effects. I couldn't help but think, could these newer technologies help us tackle problems that we had not been able to tackle before? And no problem hangs over healthcare more than the phenomenon familiar to each and every one of us: pain. Meet Professor Rob Bonin, University of Toronto Leslie Den Faculty of Pharmacy professor and co-director of the University of Toronto's Centre for the Study of Pain. Dr. Bonin's work explores the mechanisms of pain, working to discover targets for therapeutic development to dampen pain, but also understand pain from a behavioral lens.
So at a high level, we are a very basic science lab exploring mechanisms of chronic pain. So we look at how chronic pain can develop, looking from a central perspective, how changes in the nervous system may occur to contribute to chronic pain. And we also look at how that pain is expressed in preclinical models, like, for example, mice and rats. And how can we actually use these behaviors to better understand how pain affects at a higher level than just sensory or nervous function?
So how did you end up in this space? How did you end up studying chronic pain and using animal models to answer those questions?
I didn't set out to study pain. I guess my trajectory to get here might be a bit linear. I knew towards the end of high school I was interested in neuropharmacology, how drugs affect the nervous system. And so I did an undergrad in pharmacology. I did a PhD actually here at the University of Toronto under Beverly Orser studying mechanisms of anesthetics. And then towards the end of that, I was interested in or starting to ask questions about how anesthetics actually modulate pain. But what really kind of pressed me, I guess, as I went along this path, I was following questions that I found of interest, questions that I thought I was able to answer. And in that time in my PhD, there was I guess you could call it a life-changing talk was given by a McGill researcher, Kareem Nader, who was studying a phenomenon called reconsolidation. That refers to basically how memories, it's a theory of how memories can be updated to be modified or changed when you recall that memory. So at the time I was thinking about this from a fundamental perspective, saying, this should be just a basic phenomenon. This should happen everywhere. This should happen in the spinal cord. This should apply to pain. And the question really got stuck in my head, and I started looking into it in much more depth. And I just had to see if it was possible. So when I did my postdoc studies, I eventually got to ask this question. And sure enough, we found that this could happen. And this is part of what we continue to study here at U of T.
So tell me a little bit more about it. So do cells remember what they've experienced? Like, is this sort of the idea, and how does that present itself from a perspective of how we can, think of targets and drug targets that'll work on that?
In some ways, yes, the cells do have a memory in some ways. So there's a lot of, basically memories are stored in the nervous system through a lot of different mechanisms. So we know that brain cells or neurons signal to each other, and that signaling can become more efficient when cells are very strongly activated. So we call this strengthening of synapses, that point of connection, or long-term potentiation. So this is thought to be one of the first discovered or really plausible mechanisms by which memories could be stored. That you have these networks of neurons that are activated in a certain environment. For example, you put your hand on the stove, for example, and then you're going to remember that pain sensation, hand on stove, and then you not do it again. It's a one-time learning. It's a very strong association of the memory. So that memory gets stored by strengthening these connections, other cellular changes, genetic changes. And what happens then, I suppose, is that when you reactivate that memory trace, it kind of gets undone. It gets broken down a little bit, this memory trace, so that it can be updated to incorporate new information. So from a drug perspective, this has a lot of therapeutic potential. We're not trying to erase, we're not going to erase memories.
Right.
Memories are very stable. By definition, they're a very, very stable system. It's hard to modify memories. But if we can selectively tweak this process, we might be able to target those changes in the nervous system that contribute to chronic pain. So in some cases of chronic pain, for example, you can have a very strong stimulus, say surgery or osteoarthritis, when you can have this constant activation of this pain processing pathways. And in that pain processing pathways, changes can occur that are in some ways very similar to that memory formation that you would have with space or location. So what we're hoping to do is figure out how we can just activate that modifying process to sort of dampen down or weaken that memory trace of pain. So from a drug perspective, we're interested in what molecules are involved in the activation of that memory trace, what pathways erase that memory trace or change the strength between the connections there, and how can we selectively do this in a way that we wouldn't have off-target effects like, say, broad amnesia. So it's a very difficult question, and it's early days, but there's a lot of very good groups working on this now, and we have some very exciting early results.
So how do you pick the questions that you work on, and have the questions you're asking changed over the years?
Oh, very much. They change because sometimes they're wrong. So we can have this. You think what's a perfect, amazing idea, saying this has to work, and then it just doesn't turn out that way at all. So this has happened a number of times. This is part of what makes science fun, is being able to guess questions that you legitimately don't know the answer to and trying to find the answer. And when you do get it right, or you find something that's close to what you expected, it can be quite exciting.
So I'm going to say something back to you and tell me if I'm even close to understanding this. So from looking at some of your work, in some cases, you're trying to understand pain better and how cells experience this idea of plasticity. And in some scenarios, this may potentially lead to a future drug target because you figure out something that works. But in other examples that you gave, you're also working backwards, seeing how a specific molecule, you gave an example of cannabis, might also work within that. Are you kind of flipping it on its head in both ways in some of your experiments, or is it always kind of moving forward, trying to find a target versus seeing how things interact with it?
I think broadly, there's kind of two arms, I guess, of our research that we can play off of each other. And you're right that they're on sort of different ends of a spectrum of research. So one is really target identification, trying to drill down towards a molecular process or molecular target that we can hopefully find or develop some sort of drug or intervention that we can then manipulate the nervous system to dampen down pain. And on the other hand, you're right, it's a much more systems behavioral level where we're trying to look at, yes, can we modify a lot of different types of pain using a known target, a known pathway? And on the other hand, I suppose we're also trying to examine, are we actually modifying pain the way that we think that we are? Drug development in pain has a very, very poor track record for a number of different reasons, but some of which is that we use, as I mentioned, very crude or very basic models in preclinical studies to study pain, such as just evoked responses in poking animals. Is that actually indicative of pain? Maybe not. So we have to expand how many different types of pain models we have. Can we get it as realistic or valid as possible to study new potential analgesics?
Pain isn't, it's funny, they talk about this epidemic of pain that's happening around, especially in North America. And it's something that is experienced by all people. And yet we have very few really good options to treat pain. And yet we try to use a whole variety of different things. It feels like we're just kind of, as a pharmacist, you'll have a patient who could be on an opioid. They could also be on an NSAID, and then they can also be on an antidepressant. When you think about how all three of those things work, they work completely different. And yet we're just kind of guessing. Why is it that we're bundling pain incorrectly together as a symptom? Or is it that we actually just don't really have a great way to treat pain at all?
That's a really good question. And it's something I've gained a much broader perspective on since I've joined the University of Toronto Centre for the Study of Pain. One of our main mandates is to develop and foster an interprofessional, interdisciplinary approach to pain, because it's not just a symptom. And that's part of the problem. It's not just a symptom of, say, a larger disease. It's a very personal problem. Can be very patient specific. It has biological, psychological, social contributions to it, and all of these have to be addressed. So that in some way makes it very difficult to treat, but pharmacy has a very key role as well in terms of treating pain. So to come back to it, let's take the drugs that you mentioned. You said opioids, NSAIDs, and antidepressants that could be used for treating pain. So two of those are extremely old. Opioids, thousands of year old. NSAIDs have 100-year-old history. Yeah. And antidepressants, they were developed to be antidepressants. A lot of the tools that we have anticonvulsants, for example, and gabapentin that we use to treat pain. If you think about it, there's not a lot of revolutionary pain drugs that we can give. We're using hammers in some cases. We're just dampening down nervous system function to try to get to that cause of pain. Now, this, as I said, reflects it's very difficult to treat, can be very difficult to treat pain because can be systemic. It does involve, as I mentioned, a lot of different components to it. On the other hand, pain has historically been treated as a symptom of disease. And a lot of healthcare professions, even as late as even just a decade ago, didn't have much pain training in their curriculum. Veterinarians were being given 4 or 5 times more training in pain specifically than medical doctors, than pharmacists. U of T has been a leader in trying to shift that, to push more pain education, to incorporate much more pain education in the curriculum, and to, even at a policy level, really have pain recognized not just as a symptom of a disease, but as a disease in its own right. So we have leaders of our community on the Canadian Pain Task Force that have been really advocating an agenda and developing an agenda for how we can treat pain and develop treatments for pain much more broadly. So what I'm hoping in the future where this begins to go is, so you mentioned sort of this polypharmacy approach, multiple treatments. Yes, this can work because these are the tools that we have. But hopefully as we move forward in the future, we can begin to integrate that as well with other therapies, other disciplines, such as cognitive behavioral therapy, such as physical therapy, and really take a much more personalized approach to the patient.
Do you think in the future for the treatment of pain, we're going to, because you're talking personalized medicine, like figuring out the right treatment for the right patient, or are we going to actually see the biggest jump by developing new therapeutic options? Like where's the best bang for our buck going to come from in the future?
I think that's a difficult question to answer just because it is so hard to develop new therapeutics.
Yeah.
It's such a long timeline, but we are getting so much more information. We hear a lot more about the promise of AI, potentially in diagnostics. It's still not fully clear whether it's going to live up to the hype of AI, but it seems that the promise is definitely there.
Yeah.
That I think that we will be able to leverage a lot more of this massive amount of information and data mining to at least identify where we could go in terms of therapeutic development. So hopefully, at least with that information, we may get more successful on the other hand of developing treatments because we might have a better idea of where we might actually want to go instead of starting sort of from an open perspective of potential mechanisms of pain.
There's been another theme in some of the other interviews I've had around new technologies in the labs themselves, opening up lots of new questions. Are you seeing that in your field as well? Like, are there new techniques and things coming in that are just really just changing the questions you're asking or the way that you do your research?
Oh, absolutely. The past number of years, you can't, so many papers now, so many studies, conferences, presentations now, it's these absolutely massive data sets of genetics, of all the omics technologies coming together to say what is happening at a very, very high level, or what is changing in different brain regions and different spinal cord regions and different parts of the body in different pain conditions, for example, and how might these actually be contributing? How might these actually be contributing to mechanisms of pain? We've learned a lot through these types of approaches as well in terms of sex differences. We've discovered huge fundamental differences that wouldn't otherwise be predicted in terms of how, at least in certain animal models of pain, this pain develops in male versus female mice, for example. So it's these omics approaches, and of course, highly influential in all other fields, but especially in pain research as well now, just because it is such a complex problem.
With technology shaping drug development so much, I asked Dr. Anjay where he thought the future of drug development was going.
You know, there's a lot of excitement right now in gene editing and functional genomics and single cell technologies, and how all of this is helping us to identify novel drug targets, identify cell types that are important for a disease, right? And how can you target these specific cell types and not others? So I think we'll see a critical role of all of this in the next 5 or 10 years, and to furthering our understanding of how disease are happening, right? And the mechanism and the mechanistic basis of disease. And I think this is going to help us nominate additional targets for, potential drug therapies. Another big area, I think, is cell therapies, right? So developing cells as drugs. And this is really starting to transform cancer care and other diseases, right? So, but this is a very complicated process because now you need to have a completely different manufacturing process, right? How do you take those cells from patient? How do you come up with a cell product that can be administered to patient? How do you modify these cells to target a specific cell type, right? So I think there's going to be, it's already happening, but I think there's going to be a huge advances in this field, which cells to use, right? In cancer, people have used T cells, and now they're starting to use NK cells and other cells that can be deployed, if you will, to kill cancer cells. The other big area that is very exciting to me now as well is, it used to be that we talk about the druggable genome, right? And the thought that there was only a subset of the genome that could actually be targeted by a drug. This was completely challenged, if you will, or revolutionized recently with the advent of targeted protein degradation, right? So there's actually new drugs now that rather than inhibiting or activating specifically the function of a drug target, the drug actually leads to the degradation of the protein altogether. Okay, so it basically remove it from the cell. So there's been a flurry of new biotech company, I would say maybe 6 or 7, that were created in the last 2 years, right? With the idea of doing exactly this, developing targeted proteolysis or targeted degradation drugs. I think there's no such thing as an undruggable target anymore with this principle, right? You can actually basically target any gene product that exists. So this is really exciting. And this is going to require additional insight from the chemistry side of things to develop these small molecules. It's going to be also being expanded, right? So you can think that, yes, you can use these bifunctional drugs to bring an enzyme that is important for the degradation of proteins in the vicinity of your drug target. And this is how it works, right? So you have one arm of the molecule that binds to your target. Then you have the other arm that binds to this enzyme, which job is to degrade proteins, right? And by bringing them in a proximity, you degrade your target of interest. So if you can do that, you can also bring other enzyme to modify the function of a protein. Maybe bringing not an enzyme that degrade the protein, but slightly modify the function. So I see a huge opportunity there in the next few years to, advance the chemistry, advance the biology, advance the discovery of a new principle that could be applied for the development of new therapeutics. So that's a big area as well that I think is going to revolutionize our way to think about things.
I have to be honest, this episode really changed my understanding of drug development. The emergence of new technologies has changed how we continue to develop drugs. With both conversations, I couldn't help but get excited about what this means for the future of treatments. Could we see the actualization of the dream of precision medicine? The potential for this had my imagination running wild. Could we potentially see better pain treatments that are developed and personalized for each patient's experiences? Could we see a number of cancers knocked off in our lifetime? I don't know about you, but it seems to me that the future of drug development is bright. This episode of the I'm Pharmacy podcast was produced by Steve Southon, Kate Richards, and me, Mina Tadrous. It was edited by Steve Southon with musical support from Steve Southon and Diego Martinez. Special thanks to Professor Stèphane and Professor Rob Bonin for joining us this episode. We'll be dropping new episodes every month, so be sure to subscribe to wherever you listen to podcasts. Keep asking questions, stay safe, and catch you next episode.